So for this audience, for teachers, educators, this will be very let's say exotic topic, which is related to the 3D printing. We want to bring some real, let's say, not usual part, but also some interesting topic, and definitely 3D bioprinting is one of that. But I don't want to spoil and to steal your minutes. So Jakub, stage is yours. Please continue, and we can have when you finish, we can have some questions in the chat after. So just do your presentation, and we will take note about questions. Thank you.
Absolutely, thanks. Thank you for inviting me, and it's really great to have this opportunity to present. As you mentioned, maybe it's a slightly more exotic topic, but I hope that at the end of my presentation is going to be more familiar to you, and maybe you will see the potential of the technology, how you could implement it, that also in maybe your education. And I'll try to show you really that, you know, thinking in 3 dimensions is really important for technology and development of many different research fields. So I'm gonna just share my screen now, and let's hope everything goes fine. And you should be able to see my presentation now.
It's starting, but yeah, no, it's yes, we see it.
Yeah, perfect. All right. So I entitled my talk "Many Dimensions of 3D Printing" because I wanted to start with 3D printing or traditional sense that probably some of you already are familiar with, but later I wanted to continue with bioprinting. And just to introduce myself maybe a little bit, my name is Jakub Lewicki, and I'm currently working as a customer success engineer at Alevi. It's a 3D bioprinting company that produces these machines, these special 3D printers that I will show you in a few minutes. And this particular printers that we create, they're for biological applications, but I'll mention all of that. And before I started to work with Alevi, I started my journey with entire field of 3D printing and bioprinting on my PhD that I did in Karolinska Institutet in Stockholm, Sweden, where I started to research basically in neuroscience, so trying to understand our brain, different diseases affecting it. And we also started to use this technology in our lab, but I will also tell you more about how we can use that in research situation. So let's start with basically giving a short historical background on the whole field. And it goes back to 1440, where Gutenberg invented printing press. So the whole technology really revolutionized, you know, how we share information, how we reproduce that information, and that was a big step in advancing many different fields. So sharing the knowledge started to be much easier, and much later, because in 1984 there was the first desktop printer, HP laser jet printer, that was introduced. That was one of the first office printers or home printers that you could commercially buy. And this all is just beginning of 3D printing. It just started with printing something on 2 dimensions, that depositing ink on a flat surface. And this technology was taken a step further, not that long after the first desktop printers were introduced. So in 1986, this guy here on the picture, his name is Charles Hull, and he patented a machine called stereolithography apparatus. So this machine could create some 3-dimensional object from special liquids. This liquid, basically, as you can see on this image, is sitting in a special container, and the laser is being shined on the surface of this liquid. this liquid can react to this laser and turn from liquid to solid object. So we can basically, by shining the laser just on the surface of this liquid and moving the stage, as you can see on the picture, we can slowly, layer by layer, create this complex three-dimensional object. So that was the very beginning of the entire field of 3D printing. However, this technology was quite expensive at the beginning. It was quite complex, involving lasers, some optics, and so on. So soon after, there was another technique introduced called FDM, so fused deposition modeling. And that was in '91, and I show here a hot glue gun as a picture, because essentially the whole idea behind this technology is melting a plastic like in a hot glue gun and then squeezing it out in a very controlled way onto the surface, and doing that many times. So sometimes when I have presentations for schools or for kids, explaining how 3D printer works, and when I cannot take with me entire 3D printer, I just take a hot glue gun and we draw together a small pattern that we later trace with this gun, showing the principle of like heating up the material, squeezing it out, cooling it down, and then additively building layers, creating this more and more tall and more complex object. So that was '91, the FDM technology, and just a year after, there was yet another addition to the family of different 3D printing techniques, and that was SLS, so selective laser sintering. Basically, again, we had the laser here that instead of shining on liquid was shining on a powder that, because of the heat energy from the laser, could fuse together, glue together, and this way, later removing the rest of the powder, we could achieve this three-dimensional structure. So the whole field started like this, and then different applications came out in the fields. And one of them that I will focus today on is medical applications, and that's a picture from 1999. Or a bit later, but this man on the picture is Anthony Atala, one of the researchers working in the field of tissue engineering and bioprinting, and he holds a 3D printed bladder scaffold, and that was later implanted with human cells that could populate the scaffold, and later this construct could be actually implanted surgically to the patient, helping with some different diseases affecting bladder. So you can see this. There was a pretty slow development of the technology at the beginning, but later everything started to accelerate quite quickly. And when it comes to more desktop use or home or maker use, everything really changed in 2008 when RepRap technology was introduced. So this technology or this idea of printers is basically that a 3D printer can print some of its own parts. So it can kind of replicate itself, really reducing the price of the components. And that's why there is a lot of 3D printers on the market that use this idea, and you can just download some parts online for free, and then print them and then assemble 3D printer yourself. Of course, you will still need to purchase some other components, but anyway, that reduced tremendously price and entry level for 3D printing. So so how it works in general? Well, as I explained, the traditional 3D printing that I'm talking about, so FDM techniques, is basically melting the plastic and controlling it with a very precise robotic arm in 3 axes, and X, Y, and Z, and later layer by layer creating this 3D object. And for that, we need to use some kind of material. And traditional 3D printing, so the fused deposition modeling, we are using different type of polymers. It could be, for example, very popular PLA, or some other plastics with addition of, for example, wood particles or metal particles. It could be more flexible materials to achieve different quality of the 3D printing object. But most of them, they come in a plastic filament version that you can feed into the 3D printer and later use that for melting and extruding that material. And there are many different industries that found application of the technology itself. So from automotive industry or aviation, where some companies are actually using that to create parts of engines, for example. Traditional manufacturing techniques that usually start with a bigger chunk of metal, for example, and then reducing it with different milling techniques and drilling and so on. Sometimes they were not able to produce very specific and very sophisticated shapes. And here, or 3D printing can really help with that problem because you're really able to achieve complex and sophisticated shapes. 3D printing is also very popular, and for example, do-it-yourself applications. A lot of maker spaces have 3D printers. It's great, quick manufacturing, quick prototyping device. And also in medical applications that I will just mention in a second. And I also mentioned here jewelry, because with that you are able to create pieces that are not possible with normal or traditional goldsmith tools or applications. And to mention a few more real-world applications, I want to start with story. And this man on the picture is Luca Parmitano. He is an astronaut, Italian astronaut that flew with ESA crew to International Space Station. And I met him several years ago in Stockholm at a Space Explorers Conference. It was a fantastic event with 100 astronauts from all around the world, sharing their experiences and talking about research as well at the International Space Station. But when I met Luca, he told a story of his real dangerous situation that he encountered in space. So when he was on a spacewalk trying to do some routine maintenance work outside of the space station, he started to feel that the water was accumulating around his head. And as you can see on the picture, he's a bald guy. So that water in zero gravity started to form a bubble around his head. And that would be fine, but the more and more water was coming into his helmet, it started to cover his eyes, so he couldn't really see. It started to cover his ears and nose, so he didn't really hear the ground control. He was almost not able to breathe, and with all these problems, he had to make it back through to the space station. He almost drowned. They were not sure if it's toxic liquid from cooling devices or water, so he couldn't really drink it to get rid of it. So fortunately, he made it back to the inside of the space station, and he was rescued by his colleagues. I'm just telling you this story because there's so many things that can go wrong, and it's not only on the space station, but it's a great example of a remote location where it's difficult to deliver some goods. And for example, the shuttle program that is not operating anymore involves 2.5 million moving parts. There's so many things in operations like this that can go wrong, and it's not possible to prepare for all these scenarios. So what we do instead is to just bring a 3D printer to the space station, and that's actually what is happening. So on this picture, you can see 3D printed tools or parts that were created on board of International Space Station. So imagine that if there is some failure, like in Lucas' case, or Apollo 13, you know, famous case, instead of for example, trying to send a rocket with replacement parts, you could just send a file to that printer and create a replacement part or a tool that could help in this scenario. So one of really fascinating applications, especially if we start to think about traveling to more distant planets, when the travel times are going to be much longer, and it's going to be really impossible to take everything with you. Sometimes it's going to be easier just to create something on the go. And I already mentioned some medical applications, but I wanted to share with you a couple of these stories. For example, this one, this boy had his arm damaged and lost because of the wild animal attack, and in his region, prosthetics are very expensive, and also for a young boy like him, getting a prosthetic means refitting a new prosthetic every several months, because he's growing very rapidly. So to adjust the size, he would need to really have several prosthetic arms throughout his childhood and teenagehood. So to solve the problem, this group of volunteers basically created a 3D printed prosthetic arm that could be adjusted very easily. It could be printed more often, and could fit his body really perfectly, because you have all this customization possibilities when you use 3D printer. So real world example of how the technology can make something more accessible and cheaper, and really make a difference. But 3D printing can have a lot of different flavors. I show here, for example, a gun that was 3D printed. We have to keep in mind also this aspect of technology to be responsible, and whatever we create should stick to certain standards. On the other hand, we have to also think about the future, where intellectual property can be in danger, because it will be possible to, for example, download something from the internet for free, and then violate, for example, commercial rights and print something on your own at home. So there is a lot of aspects of technology and ethical aspect that we need to think about, not only in traditional plastic 3D printing, but especially in medical applications that I will talk about in just a minute. And there are also a bit more fun, maybe, applications of printing with food and creating very sophisticated desserts with different flavor combinations and crazy shapes. But also very big scale 3D printing. For example, this house on the picture was also created using really big 3D printer, using concrete, and just extruding that. You can see these layers actually on the image. These are coming from basically creating that object in this layer by layer fashion. Also, 3D printing comes into fashion industry. You could scan your food, for example, and 3D print a shoe or some inserts to shoe that will fit your feet perfectly. Also, I mentioned some fun applications. So do it yourself, making and creating toys, appliances for home, and so on. But also in more medical direction, very interesting application is creating anatomical models with a patient-specific data. So we could basically scan a patient and create an anatomical model of, for example, an organ or bone, either for education of in schools, universities, but also educating patients. So there are some programs that, for example, try to explain procedure, surgical procedure to the patient using his own data and his 3D models printed based on his or her own data, and it basically creates this, you know, better reception of what you're trying to explain, because you're really showing it on the patient-specific example, so they can understand better what's going to happen to them throughout the procedure, and so on. But it's also a very useful tool for surgeons to plan surgery, to really train and see what kind of ways of entry they have, and so on. So investigation in direction as well. But something that I wanted to talk about today as well is a very different flavor of 3D printing. It's bioprinting. And entire bioprinting comes from a tissue engineering field. So tissue engineering field is a combination of, as the name suggests, engineering and biological applications. So everything starts with a patient that has some problem. And we can collect cells from a patient, from a donor, and later start growing them in the lab. There are different techniques how it's possible, but we can grow cells outside of a human body with special machines that basically keep the conditions as they are inside of the human body, so right temperature, right concentration of the different gases. We use special liquids to feed these cells, so they feel basically like inside of a body. And this cells from the cell culture, how we call it, can be used to generate a graft, so a construct that can be later implanted to the patient. Traditionally, tissue engineering was basically a field that used any material from a household. So at the very beginning of tissue engineering, researchers would just take a foam from a mattress, a wire from bra, and try to put the cells on it, see how they behave, and implant it back to animals, and use the materials that were already available in shapes that are already available. But with 3D bioprinting, we can really take it a step further, and we can make something really custom for specific patients with materials that are really biologically relevant. So this is a good overview of 3D bioprinting technology, and I know it looks really busy, but I'm gonna take you through this slide and mention several critical steps that you need to think about when you start 3D bioprinting. So first, everything has to start with some kind of 3D model. And I said you can download them from the internet, but you can also use patient scans, for example, coming from MRI or different scanning techniques, and you can really visualize organs in 3 dimensions, create a model based on that, that is going to be used for a 3D printer to follow. And there are different ways of 3D bioprinting, as also in traditional 3D printing. Here we have different techniques. So one is inkjet bioprinting that basically comes from the office inkjet bioprinters. So in my old lab, when I was doing my PhD, that was one of the first ways to print cells. You would just basically take an inkjet printer, remove the cartridges, and replace ink with live cells or proteins or different biomaterials, and then print them on a flat surface. So that's one technology. Another is laser assisted, that has very high precision, but also prints pretty small things. And the main one that our company is working with, and I was working in my PhD, is micro-extrusion bioprinting, that is using basically a syringe with material inside that could be some hydrogel, some special biomaterial, usually soft materials. And it can extrude or squeeze out this material from the syringe, and layer by layer create 3-dimensional shape. And to make it bioprinting, we need to also include cells inside of that hydrogel. So as I mentioned, we can collect the cells from a patient, mix them with this special material, and later create this complex three-dimensional tissue model that can be used later for in vitro research in the lab, for example, looking how these cells behave, or in future also creating bio-artificial implants. And this is the machine that our company is producing. One of them, we have actually a range of different machines. This one has, as you can see, this 3 extruder print heads that allows you to print 3 different materials at the same time. It has very high precision mechanism to control all the axes. So basically, you can with really micron precision create this 3-dimensional constructs using the cells. Also, in this machine, we have a temperature control, so you can provide nice warm environment for the cells. You can also heat up the print bed, so everything sits in physiological conditions. And to print the cells, you would put this machine in special sterile chamber where there's no access of any bacteria or germs. So you can create a sterile construct that later can go in this special device incubator that maintains this right conditions for growing cells later. And this technology also comes with special attachments that basically allow you to print also special gels that react to light as well. So you have very wide range of materials and applications, including also thermoplastics. So you're not only limited to very soft tissue, but also to plastic. But in general, our body is soft, so that's what you want to use. Unless you're working, for example, with bone tissue, so then you can look into more stiff materials, trying to replicate mechanical and biological properties of the tissue that you want to create in the lab. And this is a pretty standard 3D printing software that I'm showing you here. This is open source. I just wanted to show you this workflow of having the 3D model. So this is a brain 3D model, just one hemisphere. And this is a solid object. So you have to translate this solid object into the language that is going to be understandable by the printer. And this process is called slicing. So here you can see a lot of these lines that were created by the software. So from this solid 3D model, it was the software converted it to the lines of code that would tell the printer how to move and how to behave. Basically telling it to move with given speed to given location and extrude given amount of material. So this is how the printer can convert the 3D model to actual line of commands. And later it can create objects, for example, like this one. So as you can see, some anatomical shapes, very organic shapes using soft materials, not possible to be on traditional 3D plastic printers. And there are several different materials that I mentioned. I mentioned some plastic and hydrogel materials, but there's also a different division of these materials to natural and synthetic. And there may be some familiar names on this list. For example, one of my favorite alginate that I worked on my PhD. This is really cool material also to use at schools and different demonstrations, because it's you can just buy it online or like store, and it's safe to eat. So actually, with this material derived from seaweed, from algae, if you mix it with water, it's completely transparent and it's soluble. But when you apply special salt, calcium chloride, it turns into gel rapidly. But it's not only good for a kitchen application, but it turns out it's also great for growing cells inside, because it's it's soft and cell-friendly. So I was using this material, for example, to grow tumor cells inside to do some research or on this material. And there are also some synthetic materials that are basically created in the laboratory that can be used also for different bioprinting applications. One that I wanted to highlight is actually spider silk. That's something that I was also working on during my my PhD. And spider silk is one of the best natural fibers. It's really strong, extensible, and biocompatible. And was already known in medieval ages that spider silk has this great properties of wound healing, for example. So we tried in my previous laboratory to basically use this special version of silk, spider silk, synthetic silk, to create bioprinting three-dimensional models. And because of spiders, they're not really great animals to keep in the lab because they're very territorial, we try to actually create a silk in bacteria. So instead of just farming of a lot of spiders, we just use bacteria to produce this protein for us, and later collect it, purify it, and try to 3D print with that material. There are some just details of the whole technology, but we, together with collaborators, we discovered that it's possible to mimic how spiders actually spin their web. We looked at, for example, that the different pH in their silk glands can really help with creating that spider silk line. And we wanted to apply this technology also in our bioprinting machines. And another applications that are out there for bioprinting range, again, from tissue engineering to basic research in the lab. So again, at the first picture, you can see a bladder created with bioprinting and populated with human cells that could be later used for implantation. And next, you see a sheet of skin that was also created with bioprinting technology. So these organs or these type of tissues are the easiest, or the relatively the easiest to create in the near future for real medical applications, because they usually come as a thin sheet. They have rather simple architecture. So these are the first that we expect to really see in the future for medical applications. But there are many more candidates, of course, for example, liver or kidney. There are already some successful cases of 3D bioprinting these tissues, but in a very, very small scale. And this very small scale tissue can be used for example, for screening different drugs. The current problem with the pharmaceutical industry is that it takes more and more money, and we are producing less and less new drugs. This is because the current standard method of testing medications are based on two-dimensional models. So basically growing cells on the plastic flat dish that is really stiff. It's not soft like our body. It's only two-dimensional. So it gives us a limited amount of information, and very often these models fail to reproduce what is happening in our body. That's why we need to really take it to the next dimension, and maybe not only print healthy tissues, but also, for example, tumor tissue. That's what I was doing also during my research, trying to print some tumor models, and then looking how this can react to different environments, different drugs, and so on. And actually, there are some clinical trials starting with also with transplanting ears, for example, or the constructs that can help with recreation of the ear if there was any, for example, birth defect in the patient. So just to finish it up, I wanted to show this quote from James Cameron, famous movie director, and he said that he was watching King Kong and thought that that would be really great in 3D. And everything is better in 3D, really everything. And sometimes I also think the same, that especially in research, we need to think in this three-dimensional way to answer questions that are relevant for biology and medicine. But I saw also during my years of scientific career that it's a great tool to open thinking for students and kids. We were actually hosting a lot of students in the lab at Karolinska, either high school students or bachelor master students, and sometimes giving them some simple problems to solve with 3D printing or bioprinting was really opening their creativity. And they were really excited, that they can iterate over the problem very quickly, create next prototypes, and solve some problems from basically measuring things to creating the 3D model to actually seeing it being built live. So I think this is great technology that can answer many questions. And if you have any questions right now, I'll be very happy to answer them.
Dear Jakub, thank you on my behalf and 177 registered viewers now. Maybe we have some questions. As we said, maybe it's topic a little bit exotic, but we want to include this because we believe tomorrow Greg Gage will talk about playing with the brains, with the biosensors. So those are, he made these electronics for kids. Not I don't want to spoil you his words. So, but he wanted to share with kids some of this stuff. So and experience. So I believe this is maybe now exotic, but definitely it can be inspiring maybe for a youngster, maybe to think in different way, and also maybe have idea to build some new device related to bioprinting and so on. So this is why we want, and I'm very thankful that you share your presentation about 3D bioprinting, and maybe some questions. Maybe teachers are afraid of questions about this exotic topic, but
I'm sorry, I wanted to interrupt here and answer to what you just said, because this is something really interesting, and that's how we also think as a company, that democratizing, this technology can really make difference. And when I was starting my PhD, like several years ago, this technology was super expensive. Like no one, only big labs could afford, having a 3D bioprinter like this in their lab. And then the Alevi company came with printers that were like several orders of magnitude cheaper than normal bioprinters. And that also happened with plastic 3D printing. At the beginning, only big factories could afford it, and later the technology started to be more democratic, more accessible for people. And that's also what we're trying to do. We sometimes have collaborations with maker spaces, or with we had some collaborations in the US with high schools that actually had our printers there and teaching different aspects of 3D printing, of biological applications, tissue engineering. These are fields that are really exploding right now, and it's important, I think, for the next generations of young engineers and tissue engineers to not be afraid of this, and not look at this as super exotic technology. It's becoming more and more available, and I think this is super exciting. I remember myself when I was a kid reading popular science magazines and reading about 3D printing, and I could never imagine that I would be working with this technology. But I think nowadays it's probably more and more easy to imagine that.
Absolutely, and we got that's the reason, and how I get in contact with Alevi is because last year, or yeah, I believe it was last year, we want to bring a first 3D bioprinter to Croatia with the help of US Embassy, but it was it's also you have to have idea what to do exactly with this, and have some support from our client side. So who will use it, and how to use it, and so on. So we stopped this, but this is also something what is very interesting, and I believe maybe in the meantime maybe some of the university already bought some of the 3D printer bioprinter in Croatia. But maybe we had some comments. Maybe you can read it.
Yeah, I see one question, and the question is about that I said that mixing real cells with other materials is possible for research in bioprinting, and how we could be sure that actually we get the same results from this mix as in the real tissue. Yeah, that is a great question. So basically, I will just give you a very short explanation how it's been done traditionally. So traditionally, of course, we cannot look into every aspect of disease in the real humans because sometimes it's not ethical. For example, some brain diseases, you cannot just poke someone's brain and take a piece out of it to investigate the disease. Sometimes it's just very technically difficult, and we need a bit more simplified system to look at different aspects. And that's why we created tissue culture. So basically growing cells outside of the human body in the lab. And this is not exactly, the same as in real tissue, but gives us very good estimation of what's happening in that tissue. And to compare the results, usually we make a lot of experiments on this in vitro tissue in the laboratory, and we compare it with something that we see in the human body, or with the tissue that we cut out from, for example, in the section from a patient. So we make these comparisons, and then we try to see if what we are trying to replicate in the lab makes sense or not. But what we started to see, the tissue, growing tissue in 2 dimensions in the lab is 100 years old, and not much has changed from the last 100 years. There are some, improvements, but the principle is exactly the same. And we started to hit the wall that basically a lot of the data that we collected from the standard, 2D world, they're not enough anymore. That's why it takes, as I said, more and more money to make new medications. And one of the reasons is that we started to see that cells in 2D in the lab, they don't behave like in normal human body sometimes. It turns out that sometimes just the stiffness of the material that we grow cells really matters. So as I said, our body is really soft. If you want to grow it in the artificial conditions, in the lab on the plastic, the cells will sense it, and they will behave differently. I can tell you from my experience that it's very easy to kill cancer in the lab on the plastic dish. You can put some drugs and you kill it, and you see this headlines in newspapers, new fantastic drug killing cancer. Researchers from this institute found a cure for cancer, and so on. Yeah, this is all exciting news, but it's really in many cases only based on this 2-dimensional simple models. And later, when we take these drugs to clinical trials, when we try to test them on animals and then later humans, they fail because the situation is completely different in 2D and 3D. Cells behave differently, they respond to drugs differently. there are so many more aspects, you know, of our body in this complex situation than we can mimic in the lab with traditional methods. We have many different tissue types. We have blood flowing. We have, you know, different parts that are moving and so on. So it's difficult to mimic with standard cell culture methods. But with 3D printing, you can start making more and more complex methods that have different cell types. You can start include blood vessels, for example, inside. So yeah, I mean, and still we need to validate that. So compare this 3D printed thing with a real tissue with different experiments and make sure that we are as close as possible to the real scenario. So in this way, you know, creating better models for drug discovery or answering questions about our body development and so on. So I hope it answered your question and you can understand it bit better now what is the main aim and how we try to make sure that, you know, these two scenarios are similar to each other.
Okay, thank you, Jakub.